Assalamualaikum warahmatullahi wabarakatuh and very good day to all of you. My name is Hafizuddin Kamaruddin from College Matrikulasi Sarawak. So for today, we'll be learning about genetic inheritance. Genetic inheritance, it can be divided into several subtopics such as Mendelian genetics, the types of inheritance that deviates from Mendelian inheritance, and genetic mapping. So you guys will learn about Mendelian genetics with me, which can be divided into monohybrid and hybrid. So for today's lectures, we will cover about 4 learning outcomes has been stated below. The first one will be define the terminologies used in genetic inheritance such as allele, gene, locus, genotype, phenotype, homozygous, heterozygous, dominant, recessive, self-cross, and test-cross. And then we will state the characteristic that Mendel's used in its pea plant. And also we will discuss about the Mendel's first law, which is law of segregation, and Mendel's second law, law of independent assortment. The first terminology is allele. Allele can be defined as any of the alternative version of a gene that may produce distinguishable phenotypic effect. As we can see in the picture, both dad and mom, or both father and mother, will carry their own allele, which is for father will carry a black eye color allele and mother will carry a blue eye color. So this is allele. The second terminology is gene. Gene can be defined as a discrete unit of hereditary information that consisting of a specific nucleotide sequence in the A. As we discussed in chapter 1, gene contain a lot of genetic information that are passable through generation to generation. The third terminology is locus. Locus is a specific place along a length of chromosome where a given gene is located. For example, in this chromosome, the color flower or the flower color gene is located in a specific places in this chromosome. The next terminology is genotype and phenotype. Genotype can be defined as a genetic makeup or a set of allele of organism. Meanwhile, phenotype is an observable physical and psychological trait of an organism which determined by genetic makeup. For example, in this picture, you can see that this bird, the red bird and the blue bird, have a different genotype. But in each genotype has shown a different phenotype between each bird, the red bird and the blue bird. For example, for homozygous dominant and heterozygous, both birds are having a phenotype, the red phenotype. Meanwhile, for homozygous recessive, the bird having a blue color phenotype. The next terminology is dominant and recessive. Dominant can be described as describing the allele that is expressed in a phenotype when two different alleles of a gene are present in the cell. Meanwhile, recessive is describing the allele that is not expressed in a phenotype when two different alleles of a gene are present in the cells. Means that when the presence of dominant, the recessive allele will not be expressed, while the dominant is always being expressed in each organism, especially in its phenotype. The next terminology is between dominant allele and recessive allele. Dominant allele can be defined as an allele that is always expressed when it's in present, regardless whether it's homozygous and heterozygous. Meanwhile, recessive allele is an allele that is not expressed in the heterozygous state. So as we can see in these examples, the dominant allele will be expressed, meanwhile the recessive allele is not being expressed, except in homozygous recessive, when the both recessive allele are joined together, it will be expressed in the absence of dominant allele. Homozygous is mean or can be defined as having two identical allele for a given gene. For example, big A, or small a, small a. So if it has a big A, we call it as homozygous dominant. Meanwhile, if it has small a, we call it as homozygous recessive. The next terminology is heterozygous. Heterozygous can be defined as having two different alleles for a given gene. For example, we have the dominant one, big A, and the recessive one small a. In this chapter, we will be learning two types of crosses. The first crosses is self-cross. Self-cross can be defined as a cross between male and female from the same plant of the same generation. In the Mendelian's experiment, Mendel will cross the same plant from the same generation, which is from male and female counterpart, to show dihybrid and monohybrid experiment. The second crosses that we will learn is a test cross. Test cross can be defined as a breeding of organism of unknown genotype with homozygous recessive genotype to determine the unknown genotype. So, if we do not know the plant with an unknown genotype, whether it have a big P, homozygous dominant, or big P, small p, heterozygous, we should cross it with recessive homozygous, because it will producing a different ratio or different result in these crosses. Today, we will discussing Mendel experiment, as Mendel is a father of genetics. His experiment is a breakthrough in genetic communities, especially in biology. So it's worth to understand the experiment well for us to understand dihybrid and monohybrid experiment. So in Mendel experiment, Mendel studies the inheritance of characteristic in garden peas, or we call it as Pisum sativa. Okay, he chose pea because first one, they were easy to grow. Secondly, they had a short life cycles. Third, their pollination can be controlled. Fourth, they have easily observable characteristics. So this is a pea plant and this is Gregor Mendel. This is a 7 characteristics of Mendel pea plant, such as seed shape, seed color, pod shape, pod color, flower position, and stem length. In this experiment, Mendel showed that the dominant to recessive ratio at the right side, showing that the ratio is about 3:1 ratio. The ratio is come from the number of dominant form and recessive form at the center of the table. Right now, we will discuss about monohybrid inheritance. In monohybrid inheritance, we will discuss one characteristics of Mendel's P3 plant to show what is a ratio of phenotypic and genotypic ratio that we will get from this experiment. So, in the law of segregation, states that the two allele for a heritable character separate from each other during gamete formation and end up in a different gamete. So as you can see in this picture, the allele from paternal, the red one, and the allele from the maternal, the blue one, will be segregated during anaphase 1 of homologous chromosome, and each daughter will having allele from one parent only. As you can see, it will be having allele from paternal side and allele from maternal side. Later, each gamete, for example, at the left side, the homozygous dominant parent will produce big A gametes, and from homozygous recessive parent will produce a small a gametes. When they are crossing together, they will produce a heterozygous offspring, means that they have big A and small a in one organism. Mendel breeding experiment with tall and dwarf plant is an example of monohybrid inheritance, which involve a single characteristic that determined by one gene. So the ratio of dominant or to recessive phenotype is 3:1, as been shown in a right diagram. Mendel experiment are involved in 3 steps. The first step is to produce a P generation, which is through breeding. As you can see in the figure, purple flower will cross between the white flowers that later produce the F1 generation that we call as hybrid. Later, it will be self-cross to form the F2 generation, where the ratio will be 3:1. In Mendel experiment, Mendel used a true breeding tall plant and true breeding dwarf plant that later being crossed between each other. Each plant producing a big T gamete, and for dwarf plant, small t gametes that later produce heterozygous in F1 generation that has been stated in these figures. So all genotype is heterozygous and all phenotype is tall. Later, both tall plant has been crossed between each other that we call it as a self-cross. That producing gametes, each producing big T small t, that later producing F2 generation that having this genotype, which is 1:1, and its phenotypic ratio is 3:1, as been shown in this picture, 3 tall plant and 1 dwarf plant. To sum up, as you can see from the ratio in this table, okay, when the F1 is self-pollinate, it's producing a 3:1 phenotypic ratio. Meanwhile, when F1 is self-pollinate, it's producing 1:1 genotypic ratio. If the plant is having test cross, means that the F1 plant, which is big T small t, cross with a small t, which is recessive genotype, it will producing phenotypic ratio 1:1, and for the genotypic ratio, it also producing the same ratio, which is 1:1. Right now, let us discuss about dihybrid inheritance. Dihybrid can be defined as an inheritance of two characteristics. As we can see in this chromosome, it has two different characteristics that being controlled by a different gene at a different locus. In one experiment, Mendel crossed a plant from a two pure breeding stream, which is one tall with a purple flowers and another dwarf with white flowers. So all the offspring in the F1 generation were tall with a purple flowers, which is a dominant characteristic in this plant. So when the F1 generation was self-crossed, they producing a F2 generation with the following phenotypic ratio, 9:3:1, which 9 tall purple flowered, 3 tall white flower, 3 dwarf purple flowered, and 1 dwarf white flower. What Mendel has been observed, he observed that two phenotypes resemble one or the another of the parents. As we discussed before, the 9 tall purple flowered is a dominant one, and 1 dwarf white flowered is the recessive one. And another two phenotypes has a combined characteristic of both parents, which is 3 tall white flowered and 3 dwarf purple flowered. The ratio of tall plant to a dwarf plant and the ratio of purple flower plant to the white flower plant is 3:1. This was the same ratio that occurred in the monohybrid process. So, Mendel concluded that this result that have two pairs of characteristics behave quite independently of each other, and this led him to formulate his second law. The second law is a law of independent assortment. So law of independent assortment can be defined as two or more genes assort independently where each pair of allele segregate independently of any other pairs of allele during the gamete formation. This figure shows the relationship between Mendel's law of independent assortment and meiosis. During metaphase 1, each chromosome will arrange independently. This will cause different possible haploid gamete will be produced at the end of meiosis process. Different independent assortment or different arrangement will lead to a different possible haploid gamete will be produced at the end of meiosis process. This is dihybrid table that will be discussed between F1 self-cross and F1 test cross. During F1 self-cross means that F1 cross between each other, it will be producing a phenotypic ratio as 9:3:1 and genotypic ratio that we stated in the table below. And in F1 test cross, where the F1 is crossed between homozygous recessive genotype, it will be producing 1:1:1:1 ratio. It's also having 1:1 ratio for genotypic ratio during F1 test cross.